Morning. This is Dieter Friedl. I welcome you for our webinar In Vitro In Vivo Correlations, establishing a correlation between in vitro tests and in vivo product performance. This is a webinar organized by the BPS, FIP BPS. BPS stands for Board of Pharmaceutical Sciences. I would like to introduce myself. I'm Dieter Friedl. I worked for Bayer for more than 33 years. I was in pharmaceutical development, in analytics, and in quality assurance. And in FIP, I share the special interest group, Regulatory Sciences and Quality. And I'm also the chair of the focus group, Dissolution in Vitro Drug Release. And I will moderate this session together with Andreas Abend. And I'm very happy that we have found Patrick Maroon as a speaker. As you may notice that this webinar is recorded and will be later available on the FIP website. And you may ask questions using the question box. Please use the Q&A box, not the chat for the question. This is very important later to organize the answers. And we also are very happy if you will provide feedback. And another thing is that you will also receive a certificate of attendance. And of course, you can, if you are interested, join the FIP. This is the program for this day. First, I will give a short introduction about the special interest group, Regulatory Sciences and Quality. And then we will have the modeling and simulation in biopharmaceutics, a panel discussion, which I hope will be very active from your side. And we will come to wrap up and conclusion. In the Board of Pharmaceutical Sciences, we have several special interest groups, and this webinar is organized by the Special Interest Group Regulatory Sciences and Quality. And this special interest group focuses on regulations and analytical technologies required to assure the safety and efficacy of drug products, medical devices, including consideration of bioequivalence, biosimilars, and international harmonization, with emphasis on interest and relevance to practitioners in various settings. And in this regulatory sciences and quality interest group, we have so-called focus groups. We have currently 5 focus groups. It's analytical sciences, dissolution drug release, bioclassification, biowaivers, bioavailability, bioequivalence, and clinical bridging studies. And especially this webinar is organized by the Dissolution Drug Release Group. And Andreas Abend, who will moderate later, is a member of this focus group, and I share this focus group. What we do in this focus group is we provide a global independent platform for scientific discussion among academia, industry, regulators on dissolution and drug release testing. We review and comment draft guidelines which impact dissolution testing. But also the members of this group are active in drafting guidelines. For example, some of the members were active in the drafting of the ICH guidance on bioclassification-based biowaivers, and also the group provided comment on this draft guideline. This is a quite important guideline which was recently published in 2019. And the focus group Dissolution in Vitro drug release also make some publications. I only mentioned 3 of those. That is one of the FIP guidelines for dissolution testing for solid oral products. We made a report on dissolution in vitro release testing of novel dosage forms. And also we have published position papers, because we have members from various fields, from academia, industry, regulators, and we have a position paper on qualification of pedal and basket dissolution apparatus. The focus group organizes workshops at that time before the academia. Some of you may remember that we had the opportunity to meet personally. We organized workshops on bioequivalent dissolution testing in the United States and Europe, one in nanomedicines, in dissolution in vitro release testing of novel dosage forms. And we have organized several hands-on dissolution workshops. This is a little bit practical workshops, mainly in developing countries, to improve the drug product and to teach members in these countries. During the pandemic, we organize now webinars. We have one on the qualification of dissolution apparatus and on the FIP guidelines for dissolution testing of solid oral products. And these are available on the FIP webpage. And we plan in vitro in vivo correlation, and this will take place today. And the next one is also in the planning phase, the bioequivalent in vitro dissolution testing. This will also take place in the first half of this year. And we have members from different regions and affiliations in this focus groups, and therefore we can represent quite big basket. We have people from the regulator side, from Europe, the FDA and Japan. We have people from the industry, from the United States, France and Germany, from the academia, Greece and Romania. We have from the Pharmacopoeia, from the EDQM and USP, and also one representative from the WHO. Before we start with this session, I would like to introduce the second moderator, Andreas Abend. And Andreas received his PhD degree in organic chemistry from the University of Karlsruhe in Germany. And he works at Merck Dome in Pharmaceutical Sciences department, and supporting a range of different formulations under development at his company. And he is a member of several organizations, such as the BTC, that's the Biopharmaceutics Technical Committee in the Product Quality Research Institute. And he's also a member in the Analytical Leadership Group in the Innovation and Quality Consortium. And he organized several workshops and seminars, and he is also a member of the FIP focus group. And now I would like to hand over to Andreas, and he will introduce our speaker for today.
Yeah, hello everyone, and thank you so much, Hans-Dieter, It's my pleasure to introduce today's speaker, Dr. Patrick Maroom. Dr. Patrick Maroom received his PhD from the University of Florida and in pharmacokinetics, and his career in the industry as well as at FDA spans about 30 years. So he's has been very active, very renowned speaker, contributor to the field. Currently, he heads the PBPK modeling group within the clinical pharmacology department at AbbVie. And as I mentioned earlier, he previously worked for many years at the FDA in regulatory science as an expert on in vivo, in vitro correlations for modified release formulations. And that's one of the main focuses of this talk today. At FDA, he was the head of the Biopharmaceutics group in the Office of New Drug Quality Assessment. And again, he's been instrumental in advancing the use of modeling and simulation in drug development to demonstrate virtual bioequivalence based on simulated and predicted outcomes that enabled FDA waivers of lengthy and resource-intensive in vivo bioequivalence studies. So with that, I hand it over to Patrick.
Thank you, Andreas, for this nice introduction. It is a great pleasure and honor for me to present this seminar today. I'm really thankful to the organizers for thinking about me to give this presentation. This is a topic that is very dear to my heart, as I spent most of my career involved in developing the guidance and also championing the use of modeling and simulation in drug development to reduce the number of studies that we conduct, and also to reduce the burden on drug companies that develop new drug products. I'm not able to advance the slides for some reason. Okay, so the outline of my talk, since I don't know really the composition of the audience, my talk will be both basic. It will introduce the general concepts, what we mean by the different definitions of IVIVCs, the general considerations, how you develop and how you use a correlation. And then I will give you an example of a nonlinear in vivo in vitro correlation, because most people think that IVIVCs by definition have to be a one-to-one relationship that is linear. And here I will show you an example where we used a nonlinear IVIVC and it was approved by the regulatory agencies. Also, I would like to introduce the role of safe space in this area, and how do we establish a safe space, and what role PBPK can play in establishing such safe space. And I will conclude with how we apply IVIVCs and give you some concluding remarks. Next, please. So there are 4 levels of correlation. The highest level and the most desired level is a level A correlation. It is a point-to-point relationship between in vitro dissolution and the in vivo input rate. Most of the cases it is linear, but like I indicated before, it can also be nonlinear, and it is perfectly acceptable by FDA. So this is what I mean. A level A correlation is when we, for example, we have a relationship between the fraction of drug absorbed and the fraction of drug dissolved. So this relationship allows us to convert the amount of drug dissolved into amount of drug absorbed, which we can use to predict the plasma concentration profile. And I will show you how to do it later on in the presentation. Level B correlation is when we try to correlate the mean in vitro dissolution time, and it is compared to either the mean residence time or the mean in vivo dissolution time. It utilizes the principle of statistical moment analysis. It is not a point-to-point correlation, and it is not useful at all from a regulatory point of view, because you can have many different profiles that can give you the same parameter values. And therefore, I don't think you can use it to really predict accurately the corresponding plasma profiles. So this is what I mean. When you, for example, you try to correlate the mean dissolution time in vivo to the mean dissolution time in vitro, and you get a relationship. But like I said, many different profiles could have the same mean dissolution time in vivo, and that's why I don't think it's useful. A level C correlation is usually establishes a single point relationship between a dissolution parameter, for example, the percent dissolved in 4 hours, and a pharmacokinetic parameter such as AUC and Cmax. It is very popular in for formulation selection, and formulators like really to use it to kind of develop the prototypes that they want to test further. It is very useful, like I said, in development, but a lot of times it's not useful from regulatory purposes because it does not account for the full profile. However, if you have multiple level C correlation, when you have multiple correlations at multiple time points at the different times of the profile, then in that case, it becomes useful, and you could basically get useful information from it, and also get it accepted by regulatory agencies. And what I mean by multiple level C correlation, it is, for example, a relationship between dissolution at a certain time point, at 6 hours, for example, and Cmax, or a dissolution at 9 hours versus Cmax, or 4 hours. So which means you have multiple correlations at different points of the dissolution profile with the desired PK parameter. And in this case, because you have multiple correlations, you could really assess the impact of changes in dissolution into the in vivo performance. And I think this is accepted by agencies. And this is an example where I took from a actual submission where they tested 8 formulations with different in vitro dissolution profiles, and they tested these 8 formulations into an in vivo studies, and they got the corresponding Cmax and AUC. And these 8 formulations were obtained by varying the amount of beads that are slow and fast dissolving. So which means these are this product is made of a mixture of slow and fast dissolving beads, and each profile has different amounts of beads in terms of slow and fast that will give you corresponding profile that will give you a corresponding in vivo profile. And then they were able to get a correlation from the different time points with, for example, Cmax. And this correlation was accepted by FDA because it establishes a correlation at different time points, and that's why you can really assess the impact of a change in dissolution onto the impact of the in vivo performance. And it was used both to set the specifications as well as for certain manufacturing supplements to get a change approved by the sponsor. Also, you will not see it probably anywhere, but in the USP chapter on IVIVCs, there is also a level D correlation defined. It is a rank order correlation, and it is usually qualitative in nature, and nobody ever uses it for a regulatory purpose. And what I mean by qualitative, which means that the slowest dissolution profile will have the lowest viability, and the fastest one will have the highest viability, but it is not quantitative in nature, and that's why you cannot really use it for any purpose, because it cannot predict the corresponding in vivo profile. Okay, so what do we need to think about when we try to develop a correlation? Obviously, the study design is very important, and here there is no restrictions on what type of design you could use. You could use a crossover design, which is usually the preferred, because you would decrease variability because each subject is you get deriving data for all formulations from the same subject, and therefore you will have much less variability. But sometimes if you have a very long half-life or you're not able to do a crossover design, you could use a parallel design. And I've seen cases where also they use a cross-study comparison, which means they take data from different formulation, from different studies, and try to establish the correlation that way. Usually, an inclusion of a reference is very helpful because it can be used to derive the PK parameters that you will need for deconvolution and convolution purposes. And this reference treatment could be either an oral solution, an IV solution, or even an immediate release product. And that also will allow you to scale the bioavailability to relative to that reference product. Okay, so I recommend always, if you conduct such a study, to always have a reference treatment in the study. Most of the studies are done in the fastest state because you don't want any confounding factors that will affect your bioavailability, and also the solution does not really reflect the effect of food, and that's why it is always done under fasting conditions. However, there are sometimes cases where the drug cannot be given on an empty stomach, and therefore, in this case, the study is conducted under fed conditions. And at least in my career, I've seen one case study where the IVIVC was established under fed conditions, and luckily here they were able to establish a correlation because there was no food effect on the formulation. Whether you give it fasted or fed, you always get the same profile. So that's why they were successful. Now, I'm sorry, there's a mistake in the title. There's consideration in vitro instead of in vivo. There's no restrictions from a regulatory point of view on the in vitro dissolution method that you could use. However, once you define your method and your system, it should be the same for all the formulation tested. Usually, the easiest way to do it is using a USP apparatus 1 or 2 at compendially recognized speeds. Usually, in most cases, you use an aqueous medium, either water or buffered solution not exceeding pH 6.8 is recommended. So basically, you don't need to really go into using complex dissolution method or what they call biorelevant methods to establish a correlation. If the rate limiting step in the appearance of the drug in the systemic circulation is the release of the drug from the formulation or the dissolution of the drug from the formulation, a simple compendial method would be able to establish a correlation. And we have many examples of that. Now, for poorly soluble drugs, so a lot of times they add a surfactant to solubilize the drug. However, I've seen people also attempt to develop the correlation using non-aqueous and hydroalcoholic systems. But this is usually discouraged, and I don't think regulatory agencies look at these media favorably. And a lot of the best way to do it is to obtain data from 12 units. Sometimes people try to take shortcuts and they try to do 3 units or 6 units, but the robustness of the data becomes more questionable. And that's why it's better to always use 12 units. And for you to be successful, you see a coefficient of variation in your dissolution should not be more than 10%. If your dissolution method is very variable and you have a CV of more than 10%, the probability of establishing a correlation becomes very low because of variability, and you will not be able to meet the validation criteria, basically. So having a discriminating method that is not too variable or robust, with little variability, will really increase your chances of developing a successful IVIVC. Now, how we develop the IVIVC? basically, we from the in vivo plasma profiles, one will obtain the fraction of the drug absorbed, and by using deconvolution methods. And the deconvolution methods can be either model-dependent methods, such as the Lurigan method, the Wagner-Merson method, or using a physiological-based model, or we can use model-independent methods, and these use numerical deconvolution and numerical deconvolution through convolution. So basically, the most common and the best way to develop a correlation is have the formulator make 3 different formulations with different release characteristics and find a dissolution method that will give you separation in the release rates of these formulations. Then you take these formulations and test them into a PK study, and you get the corresponding plasma concentration. As you would expect, the slowest dissolving formulation should have the lowest bioavailability, and the fastest one should have the highest bioavailability. If that doesn't work, then already that's a sign that you will not be able to develop a correlation. And from this plasma concentration profile, like I said, you can deconvolve to obtain the corresponding fraction of the drug absorbed. And like I covered this before, so basically, the way you develop the correlation is you take the dissolved profiles and try to correlate them with the fraction of drug absorbed that you got from your plasma profile by deconvolution, and you would get the relationship between the in vitro, the percent of drug absorbed with the percent drug dissolved. And that's the relationship that will allow you to predict the plasma profiles from in vitro dissolution. So this is really what we are trying to establish. What is the relationship between my in vitro dissolution and my absorption of the drug? If I am able to find this relationship, then I can develop the correlation, and then I can really predict my plasma profiles from my dissolution, and therefore I don't need to do any more studies. That's the whole concept of the correlation and its usefulness. The whole aim is to be able to predict from dissolution data the corresponding plasma profiles. And that can be very powerful because it can allow you to decrease the number of studies that you need to conduct during the approval and the development of the drug. And also it allows you to set clinically relevant specifications that are very important from the patient's point of view, because you will decrease variability for the patient from lot to lot and between lots. So basically, that the specification now can be decided not based on in vitro measures, but they can be based on in vivo outcomes or clinical benefit. Okay, so basically, if you have good correlation, whether you use a slow formulation, a medium formulation, or a fast formulation, or you lump all the formulations together, the relationship should be the same. That's usually a sign that your correlation is robust and is not dependent on the formulation that you're using. If the relationship, for example, is dependent on what formulation you use in such a case, for example, that whatever formulation I use, I get a different relationship between percent dissolved and percent absorbed, that tells me that my dissolution is not able to predict my in vivo performance, and therefore I am not able to develop a robust and predictive IVIVC. So the whole premise of the success of the IVIVC is to be able to predict, to have the same relationship between dissolution and absorption across a range of release characteristics. And here in this case, all the cases that I present are not fictitious examples. They are examples that are taken from actual submissions that were presented at FDA, or during my experience in developing drugs. In this case, for example, here the reason why we are getting different relationships between the dissolution and the absorption is because this drug is a substrate of CYP3A4 metabolism, and as we know, 3A4 is expressed at various amounts in the different parts of the GI tract, and you get different amount of absorption, and that will also give you difference bioavailability because of the metabolism. And in that case, because of this situation, you will not be able, with just a simple model, to predict the plasma profiles from absorption from the in vitro dissolution, because there are other factors besides the solution that affect the bioavailability of the drug. What other considerations you have? You need to have two formulations with different release characteristics. There is an exception to that if your dissolution is independent of the conditions of the test. For example, no matter what speed you use it, or what media you put it, the release characteristics are the same. Then in that case, you can use one formulation. And the prime example of such formulations are the OROS formulations, where no matter in what conditions you put them in, they always have the same release characteristics. Sometimes, if you have 3 or more formulations and you don't have a correlation, it is acceptable to drop the lowest or the highest and try to develop the correlation with the remaining 2 of them. And the biggest reason that one sometimes fails to develop a correlation is because we do not pay attention to how good our in vitro dissolution methodology is. And a lot of times, the in vitro dissolution that are used are not discriminating enough to be able to kind of adequately discriminate among the different release characteristic formulations. And that's why you are not able to develop an IVIVC that is predictive, that can meet the acceptance criteria. And the dissolution condition should be the same for all formulations tested in the bio study, which means you cannot, for one formulation, use one condition, and for another formulation, use a different condition. The same condition should be used across all formulations tested. And you need to basically decide on the methodology and fix it before you undertake further evaluation of the correlation. I mean, if it fails, you can still go back and refine your dissolution method and re-establish it. But you could not keep varying the conditions. You have to really have assurance that your conditions are acceptable. Otherwise, you really will spend a lot of time trying to establish it. A lot of times, if there is a lag time between dissolution and absorption, it doesn't mean that you don't have a correlation. It just means that there is a delay between the in vitro dissolution and the fraction of drug absorbed. And in that case, it is perfectly acceptable to use a lag time to correct for this. And if you use a lag time, you would be able to really establish the correlation. And the lag time can be of two types. One is just a delay, or if, for example, the dissolution is faster or slower than the absorption, then one could use also a scale factor to adjust for the differences in the time course of the dissolution versus absorption to come up with the correlation. But in these days, the softwares are very powerful that allow you to really scale it without really having to you yourself input a scaling factor. And so basically, now, how do we evaluate whether we have a good correlation or not? Basically, we have to compare whether the observed value and the predicted values are similar or close enough to each other. And basically, this exercise entails the calculation of prediction errors. And the guidance defines what prediction errors are acceptable and what prediction error leads you to conclude that your IVIVC is not predictive. And there are two types of predictive evaluation. One is what we call the internal predictability based, and this only uses the data that you use to define the IVIVC model. And the external predictability is based on additional data, test data sets, which means in the first case, you only use the formulation that you use to develop the correlation. In the second case, you use a data set that was never used to develop the correlation, like for a different study or different formulation or a different, the data was never used to really to establish the correlation. And amount of data used will depend whether you use internal versus external predictability will depend on the amount of data used for IVIVC development. Usually, the guidance and the most common practice use 3 or more formulation with adequately different release rates in the study. But for narrow therapeutic index drugs, you always have to use external predictability because of the nature of the index of the drug. And for non-narrow, if you are not able to meet the internal predictability criteria, you need to go and perform external predictability to make sure whether your IVIVC is predictive or not. And usually, for internal predictability, the percent prediction error of Cmax and AUC should not be, on average, should be 10% or less, with none of the formulation having a prediction error more than 15%. If this criteria is not met, then you need to really perform the external predictability. And usually, for external predictability, 10% or less is indicative that your correlation is predictive. 10 to 20% is usually inconclusive, and you need to really do further evaluations. And if it's more than 20%, then the correlation is deemed unacceptable. So how do we do the predictions? A lot of times people ask me, how do we really predict this solution from plasma profiles from dissolution data? And basically, this is like the concept. You have the dissolution data, you use your IVIVC model to convert your dissolution data into absorption data, and then you convolve your absorption data with the PK unit impulse function or the PK characteristics of the drug to get the corresponding plasma profile. And this is basically what I mean. You have cumulative dissolution profiles, like I showed before, slow, medium, and fast. You fit them to a model, either an Emax or a Weibull or whatever function you choose to that describes the dissolution profile. You take these dissolution profiles and fit them, like I said, fit them to a function and take the first derivative of that function to convert the dissolution profiles into the dissolution rate, which then you would use the IVIVC that you established to convert the dissolution rate into absorption rates. And then you can use a technique called convolution with the unit impulse function, the PK characteristics of the drug. You convolve these two functions to obtain the corresponding plasma profile. I mean, you could do it by hand, but these days there are many softwares that can do it automatically for you. And this is only one way to really predict. There are many different ways. This is the way I really prefer to do it. From my experiences, the easiest and most straightforward way to predict the plasma concentrations. And what we mean by internal predictability, that's another example where we had 3 formulations, and they try to compare the test versus reference formulation. And you can see that whether you use the slow, medium, or fast, all the prediction errors are met, because all of them are either 10% with none of them above 15%, and the average is below 10%. This means that your correlation is valid and predictive. And this is not really a fictitious example. This is an example taken from a submission. And that's why you see these are somewhat old graphs that I could not really regenerate. And external predictability. Here, this is basically what they tried to do is they took the data from the fasted arm of the food effect studies using the dissolution data that they had, and they wanted to see whether they could predict the corresponding plasma profile. And you could see the external predictability was very successful because the prediction error for AC was less than 10%, and Cmax was like 0.11%, which was great. I mean, this correlation was really very robust. Now, I just talked a lot about how to develop a correlation from a concept point of view and from a practical point of view. Now, I want to illustrate the actual real example that we developed at AbbVie for our formulation upadacitinib. It's a drug approved for the treatment of rheumatoid arthritis, and it's a controlled release formulation. And what we did is we tried to develop the correlation using 4 formulations where we tested them in vivo, as you can see, and then we got the corresponding plasma dissolution profiles, and then we deconvolved them and got the corresponding fraction of drug absorbed, and we correlated it with the in vitro dissolution profiles. And all this data was published a couple of years ago. Unfortunately, we were not able to develop a linear IVIVC because if you see the relationship between the fraction of drug absorbed and the fraction of drug dissolved is nonlinear in nature. So which means the relationship between in vitro dissolution and in vivo looks something like that, okay? Oh, sorry. Looks like something like that. So everybody says, oh, you don't have a correlation. But no, the correlation exists, but the relationship between in vitro dissolution and in vivo absorption is not linear. It follows this scaling factor, and it was validated and was reviewed by, in fact, all agencies around the world, and they all found it acceptable and approved it. So basically, my message to you is, even if you don't see a linear relationship between in vitro and in vivo correlation, it does not mean that you don't have a correlation. It just means that you might need to use more involved models to develop the correlation. And many times it can be nonlinear, and it is perfectly acceptable. And here, this is the data that we got. We tested the target formulation was target C, and then we had different formulations with different levels of HPMC that we tested in the trial. So we had a 10% HPMC level, 20%, which was the target. We used a 15% and a 35%. So we had 4 different formulations, and we also had a reference formulation for deconvolution purposes. And basically, as you could see, that we were able to show that our prediction error was always below 10%. And that's why the IVIVC was deemed acceptable, even though at first glance we say, oh, you don't have a correlation because it's not linear. But this is a perfect example where a nonlinear IVIVC was developed and accepted and was used not only to set the specifications, but also to get some manufacturing supplements approved by the agency, by FDA mostly. It was a process change and a manufacturing site change. And usually this type of supplements would require for us to do a bio study. But because we had an IVIVC, because we had the IVIVC, we did not have to do any bio studies, and we got the supplements approved just based on the predictions of the IVIVC. Okay, so now I talked really mostly about the concept of an IVIVC, and it is really a quantitative relationship between the in vitro dissolution and the in vivo, which means that it's a numerical kind of correlation that allows you to really predict mathematically the relationship between in vitro dissolution and in vitro absorption, which will allow you to predict the plasma profiles. Now, what happens if, for example, you don't have the ability, or you don't find that you have a good correlation between in vitro dissolution and in vivo correlation and in vivo performance? It is possible to kind of define what we call a safe space. And what I mean by safe space is the boundaries defined by dissolution profiles within which drug product variants around the target are anticipated to be bioequivalent to one another, which means you can really establish what we call a safe space by defining the range of dissolution profiles where your product is still bioequivalent. And it can either be constructed in two ways. One is by using actual data, which means you perform a study and then you determine what release rates lead you to a bioequivalent profiles, and then and the range of dissolution rates between these rates is going to be bioequivalent, and this is what we call the safe space. But now, because in the recent future, because we have advancement in the field and the capability of mechanistically understand the relationship between absorption and plasma profiles, and because regulatory agencies are relying much more on modeling and simulation, we can also use PBPK modeling, where we can really use the model to predict the range of profiles that are predicted to be bioequivalent. And most of the times, the safe space approach is used for immediate release formulations, where the release of the drug from the formulation is not thought to be the rate limiting step in the absorption of the drug. So which means this can also be another powerful tool to really use to decrease the number of studies, even though it is not a mathematical relationship, but it is very useful because it can define for you the range of release rates where you are safe, where you're going to be bioequivalent. And as long as you stay within these ranges, you will always assure bioequivalence for your products. And basically, you could use what we call the mechanistic ADA model for deconvolution purposes, or for establishing the safe space. And I'm going to give you an example that we use internally within FV. It is a drug called Elagolex. It is proposed BCS class 3, but it doesn't really meet class 3 100%. It is high solubility but low permeability. So we developed a PBPK model for the purpose of to support recommendations for drug interaction potential. But it does mean that the PBPK model can only be used for clinical pharmacology purposes. We were able to use the model to also for biopharmaceutics purposes, which means we developed a model, a general PBPK model that can be used for many different purposes. And we validated the model to make sure that it is predictive of the actual data. And then we use modeling and simulation to really define different scenarios. And basically, we kind of verified the PBPK model that is able to predict the actual plasma concentrations. But because regulatory agencies do not have any standard criteria for the acceptance of PBPK modeling, PBPK models, like what we have in IVIVCs, and the IVIVC guidance, there is a clear criteria where you have to meet for you to be able to accept the model. Here for PBPK modeling, there are no defined criteria. So internally, we decided if we are able to predict the plasma concentration within 25% deviation from the mean, I think the model will be accepted. And that's what we adopted. And in fact, the agency agreed with us. They did not question our criteria for the model. So then, how did we use that PBPK model? We use it twice within the agency, within the submission. We got our specifications approved, but during commercialization of the product and scale up, we started to see a trend in slowness of the profiles compared to what we tested clinically. We still were meeting the specifications, but we did not want to take any risk. So what we tried to do is we tried to kind of amend the specifications by doing some modeling and simulations, trying to simulate a profile that is slower than what is the recommended specification, and see whether this profile results in a bioequivalent performance. So basically, we used the model, we basically amended the model to incorporate the dissolution as the input into the model and predict the plasma concentrations that correspond to these various dissolution profiles. And we basically were able, so basically what we were, we used the different profiles. See here, so we had different dissolution profiles, and basically we were able to establish safe space where the solution will still be resulting in bioequivalent performance. And basically, based on the fact that the new proposed dissolution profile is bioequivalent to the reference which was tested in the clinical trial, we were able to convince FDA that our new proposed dissolution spec is still bioequivalent. Okay, so basically, we basically defined the range where the profiles were bioequivalent, and based on that, we showed that the new profile is still bioequivalent. The new specification will not allow any formulations that are deemed not to be bioequivalent. And basically, that's basically the safe space that we defined, the area shaded in the green. So which means as long as the profiles stay within that range, we are still bioequivalent. And therefore, we do not need to do any bio studies to approve a manufacturing change, or even to amend the specifications, or whatever regulatory purposes we need to do that would otherwise require us to develop, to perform a study. The second example is that we had to bridge the clinical formulation with the commercial formulation. And basically that would have required us to do a big bio study involving more than 200 subjects. And I think this was a quite burdensome endeavor, because it would require a large amount of subjects to recruit, which is very difficult. And also it will be a costly study, costing over $6 million to perform the study, basically. So what we did, we used the same PBPK model to kind of show that the prototype formulation that we have, that we are proposing to commercialize, is still bioequivalent to the phase 3 formulation that was tested in the clinical trial. And we showed that from our PBPK modeling simulations, that the two formulations are bioequivalent. So basically what we did is we established what we call virtual bioequivalence using a PBPK modeling approach without conducting the study. And we're able to waive the requirement of the study, and FDA agreed with us, and they approved that formulation without really having to conduct any additional studies. I think that was a major achievement, because that's something novel that we did, that we were able to approve a formulation without having to conduct any studies, and we saved the company time and money, and also we were able to not expose subjects to unnecessary studies when we really can determine the outcome of the study by modeling and simulation. And basically, this is what we did. We were able to show that these formulations are bioequivalent, and because they are predicted to be bioequivalent, there was no need for us to develop to conduct the study. And this is basically somewhat equivalent to the safe space approach, because at the same time, when you show virtual bioequivalence, you at the same time also establish a safe space. And this is what I described here to you. So basically, if you use modeling and simulation and you develop a correlation, you are able to really use in vitro dissolution as a surrogate for bioavailability. That's the whole concept, that dissolution will becomes your surrogate for bioavailability, which you can use to predict the plasma concentration profiles that will allow you to decide whether you need to conduct a study or not. And by enabling the solution to become a surrogate for bioavailability is very powerful, because it really helps you decreasing the number of studies and also save you a lot of time. And so what type of waivers one can obtain by when you have a correlation, or you establish safe space, or use PBPK modeling for virtual bioequivalence? Any type of waiver could be used for manufacturing site change, equipment change, method of manufacture, source of raw material, formulation changes. So it's pretty powerful. So it decreases the number of studies that you need to conduct, and therefore it decreases your regulatory burden. So what are the criteria for you to be able to be granted a bio waiver? So basically, if you have the test formulation, and now you do a change, and you have the corresponding, you have the reference formulation in black, and then you do a change to your formulation, and you get the yellow line, and basically, so this is the dissolution difference between the two formulations. You use your IVIVC to predict the corresponding plasma profile. If the difference between the predicted and observed plasma concentration profile is within 20%, then you don't need to do a bio study, because these two formulations are deemed to be similar, and no further testing is required. If the predictions turn out to be beyond 20%, then you will not be able to obtain the waiver. And in this case, I would not even recommend to conduct the study, because the study would fail. So, which means that you would need to really go back and reformulate or see what caused the test to be different than the reference. So, in conclusion, IVIVC can impart in vivo meaning to the in vitro dissolution test, and thus improve the quality of a controlled release product. Not only a controlled release product, but any product if you have the relationship between in vitro dissolution and in vivo performance. And firms are strongly encouraged to attempt IVIVC and use modeling and simulation to really understand and define the ranges where they are bioequivalent, because it really is very helpful, not only from a development point of view, but also is very helpful from a maintenance point of view, because you decrease the number of studies that are required to maintain and support the product on the market. Like I said, and a lot of times, I did not discuss that too much, but the solution these days is not only, the solution specifications are not only based on the in vitro considerations, but regulatory agencies expect you to have what we call clinically relevant specifications. And without understanding the relationship between the in vitro dissolution and the in vivo outcome, you will not be able to establish a clinically meaningful relationship. So which means that you have to use modeling and simulations and establish IVIVCs to be able to really put the proper and acceptable dissolution specification in terms of from the agency's point of view. I think that's all what I had. I thought I had 1 hour, but we have some more time for questions and answers if you want. Thank you very much for your attention.
All right. Well, thank you, Patrick. Really, really very comprehensive overview of IVIVCs for extended release products. There were actually a number of really good questions on the Q&A board, and why don't we get started with some of these? There was again, very simple question that you mentioned earlier about the choice of media for this dissolution testing. And why are non-aqueous and hydroalcoholic systems discouraged? I think for extended release, I can know why, but I'll let you answer that.
Yeah, I mean, hydroalcoholic solutions or organic solvents are really very harsh on the formulations, and the probability of having a discriminating condition becomes very, very low, you know. So that's why they're not encouraged, because the probability of success is very little. And I'm not saying that you cannot use them, but in that case, you can use them, but it will require a lot of justification where you have to go through a very unusual, harsh conditions, because they really destroy the integrity of the formulation, maybe.
Yeah, I'm just thinking about, you know, if you have film coatings that are actually controlling the release mechanism, you're going to be dissolving those directly in your alcohol, so you're not getting.
Yes. Yes, it's not meaningful basically anymore. Yeah. Yeah.
And there was a question about the failure of establishing a discriminating in vitro method. So frequently the discrimination relies on low variability of the dissolution profiles. Again, I think the coefficient of variation less than 10% is usually something that the agency wants to see. Maybe at the early time point it's 15%. But while the variability of the in vivo data is much more important from patient to patient. So it's that variability in vivo versus the in vitro requirements. Are we expecting too much from the dissolution methods?
No, because even put aside on the aside the question of IVIVC, if your dissolution method has a high CV, more than 10%, the agency will question the validity of your method. So whether you're using it for IVIVC or not, they do not expect you to have a very variable dissolution method. So even as a quality control tool, you know, you cannot have a highly variable dissolution method, you know. And if you adopt a dissolution method that is very variable, you are really penalizing yourself, because you will, in some instances, reject lots that are very good because they might not meet the F2, or they might fail some criteria just because of variability. But when in vivo, it is not different. So basically, dissolution tends to be over discriminating compared to in vivo, and that's why you need to minimize the variability in the performance of the method.
Yeah, I agree. I mean, it's always this threading a needle or a line into a needle. It's can be very challenging. So,
it's a fine balance that you have to kind of balance between the discriminating ability of the method versus the amount of variability that you have.
Yeah, and that's why these in vivo studies actually become very important to actually justify the dissolution method conditions.
Yes.
There's a question about, can two formulations be used to establish an IVIVC?
Yes, I addressed that in my talk, you know. two formulations can be used. They have to be different in release characteristics. And you have to also go through what we call the external predictability, which means if you use two formulations, no matter what, you always have to externally validate with a different data set, the correlation. While if you use three formulations, you don't need to go through the external validation. So yes, it's possible. And I've seen it done I've seen the IVIVC developed with just two formulations. But the range will be much narrower. The range of release rates where you can use the IVIVC is much narrower compared to if you use 3 or 4 formulations.
Yeah, because at the end of the day, the requirements for the dissolution profiles is that they are dissimilar, meaning they have to be, again, as judged by F2, they have to be meaningfully different. So
at least 10% in means, yes.
Yeah. So that's why I would probably discourage using just 2. As you said, that gives you a very narrow space.
Yes.
Can an IVIVC be employed on different formulations? For example,
No.
tablets and then capsule?
No. The IVIVCs are formulation specific and also product specific, which means if I'm company A developed a correlation for a product, another company cannot use my correlation. It is proprietary. So whatever I develop, I'm the only one that is able to use it. And it cannot be, even let's say if I developed a correlation for tablet A, I'm a different company or the same company trying to develop a tablet with a different composition or different platform, I need to reestablish the correlation for that new tablet. So they cannot be interchangeable now.
Very good. There was another question with regards to the safe space, right? So, yes. Well, I'll just read this to you. We had a case in the past that one formulation, a powder in the bottle, was dissolving very slowly, about 30% within 120 minutes, while the IR tablet was showing complete dissolution in 90 minutes. Both formulations were shown to be bioequivalent based on a bridging study. Can we consider these two extreme profiles as our acceptance criteria for dissolution testing? And is that considered clinically relevant then?
Okay, see, and there is a controversy even within FDA. Some reviewers, in fact, that requires you to establish a safe space with the same type of formulation, which means you have different variants of the same kind of dosage form, but different release characteristics, and you establish a safe space. Other reviewers, including myself when I used to be at the FDA, were more permissive, saying no matter what dissolution data, no matter what formulation you use to generate the dissolution data, as long as you define the release rate by which you are delivering the drug, you could establish the safe space. I mean, I don't think there is one way or the other. It depends. It's not. Most recently for Elagolix, one of the reviewers forced us to develop data using the same formulation. So
it's actually an interesting question that while I read it again. So you have a powder that dissolves very slowly, yet the tablet is fast. So in other words, if you just think about dissolution in a stepwise process, right? So you have film coating that dissolves, and you have the tablet disintegrating, well, you're already in a powder. So those steps don't even happen. I would probably want to know, what is the dissolution method actually measuring? Is it measuring the API? Is that truly the rate limiting step here, or is it something else? But I'm glad to see that the agencies are open to. Yeah,
but I can tell you in that case, the mere fact that the dissolution of the tablet is slower than the powder tells me that that's the reverse, and which means that there are solubilizing excipients that are making the API dissolve faster, and the probability of the agency to accept such data will be less, I would say.
Yeah, thank you, Patrick. There was a question about the software, which software is suitable for IVIVCs? I think you already talked about it, but
I mean, there are many commercial software. I even developed my own equations and my own. I did the IVIVC on an Excel spreadsheet, believe it or not, you know. So there is no, the most common one, I don't want to do some PR here, but I've seen the most common one is used by Cetera or the Phoenix. It used to be called the Phoenix, but also there are other software that are cheaper and as good, you know. There is, like, for example, the Puma's, you know, software that can let you do the IVIVC. There is also a software developed in India called IVIVC Express. It's very cheap and does the same thing also. So I mean, there is no limitation as long as the software is validated and is acceptable and gives you a reproducible result. It's okay, you know.
Yeah, I think, I mean, my understanding is that the agency, the FDA at least, will reproduce your IVIVC anyway. So
no matter what, they will ask you for the control files and they will rerun it themselves.
Exactly. So there will be, this is not just relying on the applicant's data and reports. Yes, so how acceptable is IVIVC to the regulatory agencies? I think this is probably more from a global perspective. And are biostudies necessary after IVC studies?
IVIVC, okay. Yes, regulatory agencies are very open. Some, I think Japan is the only one still in the stage of trying to explore it and develop it. Brazil in the past used to be not very receptive to that, but I think more recently they are more open to that. So in our UPA program, which was approved 2 years ago, it was accepted by each and every single agency in the world. So it is pretty accepted, even though the IVIVC was not the usual IVIVC, because it was nonlinear, that needed a lot of transformation, and we need to do a lot of explanation just for the reviewer to explain what we did, because it was a more complex modeling effort. You know, it's not the usual method, but it was successful, and at the end they accepted it. So it is quite accepted. When you need to do an bio studies, if you change the formulation in such a way that you go beyond the ranges that you tested or you develop the IVIVC with, and you don't know whether the IVIVC is valid in that area, you know, if the release rate, for example, is outside the release rate that you tested in, with the IVIVC, then you need to maybe establish the correlation, you know, to do a bio study. Or if you fail the predictions and you are still convinced that you have a bioequivalent product, you could conduct a study, but that means that you don't have a good IVIVC, basically. So I very seldom seen a case where people had an IVIVC that they went ahead by themselves and did the bio study to do the bio studies. Sometimes some regulatory agencies, very few cases where an IVIVC is accepted by one agency and not the other. So the other agency might require you to do bio study, then you go and do the studies. But you wouldn't do it by yourself. You know, only if you were prompted by the agency to do the bio study, you would do it. But it's very, very rare cases, I would say.
Yeah, it's not something like, you know, if you have like a predictive model, let's say you have a PAT model that you deploy, you know, in manufacturing, and you have to make sure that the model is still valid and you maintain that model. Once you have established an IVIVC, usually that's all you have to do.
Yes. Okay. Yeah, sure.
There was well, let me skip this one question because we'll save that for later. That might take a little bit longer. But may I know why PCS class 1 drugs are not suggested for convolution method in IVIVC?
Because by definition, if you meet the BCS class 1, you don't need to do any bio studies. And the solution, for you to have a successful IVIVC, the rate limiting step should be the dissolution or the release of the drug from the formulation. By definition, BCS class 1 drugs, dissolution is not, because it's very fast dissolving, dissolution is not the rate limiting step in the appearance of drug from the formulation. So that's why you don't need to really, and also IVIVCs are mostly for modified release formulations, and the BCS classification is not accepted or valid for modified, it's only for immediate release dosage forms. So that's why whether you have 1, 2, or 3, if it's a modified release formulation, you cannot use the BCS classification to get a waiver. It's only for the immediate release formulations where dissolution is not the limiting step.
Yeah, thanks. That's a good clarification, because a lot of times people apply solubility and permeability of the drug substance to the BCS class, but it's really
it's a product.
It's
product dependent and dose dependent. Sometimes at one dose you are a BCS class 1, and if you go to a higher dose, you don't meet the criteria anymore. So
BCS class 2.
yeah, exactly. So
yeah. All right. So for the in vitro dissolution testing, should this come from 12 units, and how many in vivo data should there be that is enough to make a good model?
Okay, the 12 units is a regulatory requirement that you would see in the IVIVC guidance and in dissolution guidance. And also when you meet with mostly with the FDA, they will always ask for 12 units. Sometimes if your dissolution is very robust and you don't have a lot of variability, they might let you get away with 6 units, but not less than 6 units. Now, from an in vivo point of view, it all depends on the PK characteristics of your drug. I've seen studies as low as 6 subjects and as high as 48 subjects. So there's no magic. It depends on the PK characteristics, how variable the formulations are and the PK performances. The more variability, the higher the number of subjects. 6, I think, is 6 is way, they are lucky, that by fluke it happened to them, but typically it's 12 to 18 subjects that usually. And our UPA, we did 24 subjects.
The prediction errors are likely going to be very large, so that's why you need to make sure you have enough. I mean, it's
a question of precision, how precise you want to be, really.
Exactly. Okay, so there was a question actually related to your IVIVC in the example that you explained. Did you try a power of time function for a nonlinear IVIVC?
We tried 14 different scenarios, and we thought that this is the best one to come up with. I'm sure there might be other functions or other mathematical scaling that will also be successful, but in our case we deemed this one to be the best. Like I can tell you, we tried maybe 14 different ways before we tried 14 different conditions in vitro also. So we did quite a bit of work, and we were insisting to be some other company would have said forget it, it's not worth it, but we believe that it is to our benefit to have an IVIVC, and that's why we put the extra effort. And it paid off because we were able to obtain waivers for a couple of supplements. So yes, you put it with more extra effort in the beginning, but you will benefit from it later on post-approval also.
Yeah, I think that's what people always seem to forget. The value is in, once the product is on the market. We move products from to different sites. There are always changes. I mean, you will not be able to source the same excipients, API, etc. So it's really important to think about this. There was a question why only the dissolution testing is used as a surrogate for biowaiver studies. Are there any other, I guess, in vitro methods for that in the future?
I mean, so far dissolution is the most common. It doesn't mean that in the future there might not be other in vitro technologies that will allow you to predict plasma profiles. But to date, the most meaningful in vitro test, which is an integration test, basically, because it takes many different factors, because many different factors could affect the solution, and that's why it's pretty predictive, But I could, with maybe technology in the future, who knows, they might come up. I mean, even the ICH guidelines, if you are able to correlate this integration with the solution, you could use this integration. but I've never seen it done really, because it's very difficult.
Yeah, we will see probably more of that when, as companies go towards continuous manufacturing, probably real-time release testing, and seeing whether or not they can use that data to actually justify that those changes that may have to happen do not impact quality. So that's really an exciting, field going forward. There's a question about different batches of the same product. Can different batches of the same product be compared to the innovator? Well, I think so.
Yes, I mean, for what purpose?
Yeah, exactly. I mean, it's quite interesting, maybe, again, and this is not available to a company that is developing maybe a generic version of an innovator drug, but the regulatory agencies probably see different generic, applications using the same drug substance, going after the same, therapeutic benefits, but they may be different. But at the end of the day, yes, there will be bioequivalence to the reference listed drug. So at the end of the day, different formulations, processes can lead to bioequivalent materials. That's by no means any difference. It would be nice to actually see this, because I'm interested in this, because I think, especially for immediate release drugs, many times running BE studies is not necessary, because there is a relatively, there's much lower risk of not meeting PK or not meeting bioequivalence for immediate release drugs compared to modified release. I think because in modified release, you're purposefully modifying the release to meet a certain release profile. What are your thoughts, Patrick?
I mean, running different batches. I know generics, I think they in terms of dissolution, they require them to do 3 batches, but not from the in vivo point of view. But I don't see in our company, we always have 3 validation batches. So we always compare even ourselves to ourselves, so make sure that our process is consistent. But in terms of generics, I don't know. You can always pick sometimes you can pick something by chance and not be bioequivalent. So it's a difficult question to answer, really.
Yeah, again, that's probably case by case. Why do there's a question, maybe that's a misunderstanding. Why do we ignore the lower release profile and the higher release profile when doing the comparison for in vitro, in vivo correlation?
Okay, I can answer. Any correlation will only be valid over a certain range of releases. So sometimes, depending on how you choose your release rates that you test in vivo, the fastest one or the slowest one might not be within that range where the correlation is valid. So which means it doesn't mean that you don't have a correlation. It's just because you extended the range of the correlation too wide, So which means if you remove the slowest one or the fastest one, you might still be within the range where the IVIVC is valid. And that's why we do it that way. You cannot really remove the one in the middle, because then you don't have, it will not help you out, So that's why we do the slowest or fastest one.
Good, excellent. I think there was this interesting question, because this may be more from a regulatory, may have a different aspect of product quality. Can an IVIVC be applied in case of difference in source of material for the same formulation? So whether you go, let's say you have hard gelatin capsules that might have different origins. So we typically avoid bovine types of gelatin capsules. Porcine origin could be not acceptable in some markets.
Yeah, technically, it changes in the source of the raw material does not trigger a biostudy usually, as long as you meet all the requirements of that raw material. So which means if all the specs are met and that for that raw material, there is no requirement to conduct a bio study. And dissolution is meeting the specifications, you're done. You don't have to do anything. You don't even have to compare, you know. But many times, a change in raw material sometimes trigger a change in process. Okay, and if you have a change in process that would require you a biostudy, then you would do the biostudy. But a simple change in the source of raw material for an excipient that really will not really affect you, there is no bio requirement.
Yeah, this is more a question of, again, is it acceptable to use that material in a certain market? So
yes, yeah, but I'm talking about FDA. I'm not sure about the requirements outside the US, because most of my experience is US based, but sure. Europe is similar to FDA, I would say. So
okay, so we have 5 more minutes. Dieter, is there any, should we stop? Are we going for, you know, final conclusions, or should we have maybe another question?
Well, we can continue the Q&A session. I think we have not so many questions anymore.
Yeah, there's 3 more questions left here, so maybe start with this.
Why don't we answer them? So like that, these people won't feel left out, you know.
Yeah.
Okay, so let's go.
Continue with the Q&A.
Yeah.
There was one question I skipped, and that was about the in silico part. I guess we can talk, you know, for hours about the in silico, you know, how you develop an IVIVC in silico and how do you run virtual VE studies in silico. So those are things that require a lot more, but maybe there was one question in the last slide with the graph. The attendee observed, seeing the study formulation was at a high level to the reference formulation. I would like to know at what percentage is it allowed to drop as to waive the VE study?
What do they mean at what level? I mean, if you are within the 20% range, you're okay, you know. Yeah. Or you can even get even simulate a stricter criteria meeting the confidence interval, you'll be okay. As soon as you are exceeding 20% difference, that's where the problem will occur, you know. I don't know if I answered the question because I didn't understand the question that well. And I mean, we had 2 different strengths of the formulation, and we did the simulations on both strengths, and both of them gave the same results. So the strength really, if you have linear PK characteristics, I don't think the strength is going to make much difference.
Yeah, it's really the prediction errors and
yes.
how you build your model.
Yes.
How do we account for patient variability in an IVIVC? I think looking at special populations or indeed variability within a population.
Yes, when the IVIVC guidance was developed, we wanted to make it simple. We just saw all the decision making is based on mean data. If you notice, bioequivalence is based on confidence interval approach, which takes into consideration the variability. The IVIVC is a bit more permissive because it allows a 20% difference in mean values, so which means it ignores any potential variability to make it a little bit more useful. Now, if you use a mechanistic PBPK modeling approach, you could also incorporate any patient characteristics into the model and also incorporate the variability into the model and come up with it. Even for IVIVC, you could develop the IVIVC with much more complex models that also accounts for variability. And also, when you do the predictions, you can use a Monte Carlo simulation approach where you can really come up with confidence interval. But these are very intense modeling techniques that for the purpose of the guidance, when I was at FDA and was part of the working group, we did not deem it necessary because we wanted to encourage the concept of people using that concept for waiving studies. So we wanted to make it simple, you know. Now that's a fault in the approach, but it worked fine so far, you know. So, but it's possible to account for variability in the subjects by using more complex. Even you can use a more population approach in IVIVC if you wanted to.
Yeah. Is it possible for the IVIVC to be done on different strengths of the same dosage form?
Yes. As long as your PK is linear again.
Yeah.
And the composition, and then the different strength use the same release mechanism for the formulation. If one strand uses a different platform compared to the other strand, then I don't think that would be, because the IVIVC is really release mechanism specific. Mm-hmm. So you cannot really mix and match different release mechanisms.
Yeah, and it would be interesting to see if the different strengths also have, if they're just dose multiples, if you're going to see differences in dissolution profiles, because that's the.
I mean, we developed, for example, in our case, we developed the correlation with the 30 milligram strands, but we went back and did the evaluation of the 15 milligram, which means we tried to take the IVIVC that we developed with the 30 milligram, and so whether we could predict the 15 milligram to make sure that it is valid across the different strands. And yes, it was acceptable. But I could see certain scenarios if you have nonlinear characteristics of the drug, and you have big differences in the formulation between like 30 and for example, let's say we go to 50 milligram or 100 milligram, and we have to use a different platform, then it becomes a little bit trickier, you know. So, but yes, as long as it is the same type of formulation and you have linear PK, it is valid across all the different strands.
Okay.
And you can show it, and you can show it also.
Yeah, awesome. Yeah, this was the last question. So thank you, Patrick. You really, passed your exam here.
Okay, thank you. I hope I didn't go too fast, because I had a lot of material. And believe it or not, I cut so much of my material. I could talk on this for like 5 hours if I wanted to. I have so many examples, so many scenarios. But I just wanted to give the concept. I want to introduce the people to the concept and its usefulness and its the future. Regulatory agencies are going to expect from you to have understanding of the relationship between the solution and the in vivo performance. It is not like the old days that I do this, the solution is a checkbox, and that's it, I'm done. You have to do much more than that. We are in a different era now, which is beneficial to the patient because we are understanding the impact of changes in these characteristics on the in vivo performance and the clinical performance. So the patient will get better, less variable products, for the patient.
Yeah. I would like to thank you, Patrick, very much for this excellent presentation and answering all the questions.
No problem.
Andreas, thank you for the moderation of the session. And also, of course, the participants for attending this meeting and the good questions, so that we had a very good discussion in this session. And I would be very happy to welcome you again to our next webinar, which will be on the biorelevant dissolution testing. And FIP will announce when this will take place. And I'm very happy if you could join us again. And I would like to thank you all and have a good day, or even we have different regions, and stay healthy.
Thank you.
Thank you again for inviting me.